Today, I will be discussing DNA replication in prokaryotes by using an analogy related to the split ends that grow in your hair. There's lots of proteins involved in DNA replication. Here's a rundown of the concepts and proteins in my analogy. Prokaryotes have circular DNA that undergoes semi-conservative DNA replication, meaning it will conserve one strand while also adding a new strand. Initiation of replication requires an origin of replication, which is a particular DNA sequence that the initiator protein will bind to in order to cause the DNA to unwind. The origin of replication is represented by the location of split end growth. Because DNA is plectonic, it needs to be unwound first before it can be pulled apart. For example, curly hair cannot be separated into individual strands easily without the use of different products or methods of separation. So the initiator protein, or a knot in your hair, attaches to the origin of replication, causing local unwinding of DNA and a short section of single-stranded DNA. Now we are beginning to unwind the DNA. An enzyme called helicase, or as a hairbrush, attaches to the replication forks and breaks the hydrogen bonds that are holding the two strands of DNA together. Single-strand binding proteins coat the single-stranded DNA to protect and stabilize it and to prevent hairpins. This is represented by detangler, which protects the hair. Gyrase is a topoisomerase that relieves supercoiling of DNA by moving ahead of the replication fork or beginning of the split by nicking DNA to reduce torsional strain and release tension. We're going to go look at one side of the replication bubble more closely. Remember that replication is occurring in different directions on each strand, and that DNA is synthesized from the 5' end to the 3' end in that direction. The leading strand is also known as the template strand. It goes through continuous replication in the direction of replication fork. The lagging strand is also called the coding strand. This strand synthesizes in the direction away from the replication fork and is a discontinuous process. This discontinuous replication creates short lengths of DNA called Okazaki fragments. This strand has to wait for more of the fork to be available for the synthesis to occur. We're going to look at one side of the replication bubble more closely. RNA primase autogenerates RNA primers to bind to the 3' end of DNA. RNA primers act as a binding site for the start of replication. DNA polymerase III synthesizes DNA by adding nucleotides to the existing primers. It begins with the free 3' hydroxyl group on the last RNA nucleotide of the primer. DNA polymerase I uses 5' to 3' polymerase activity to replace RNA nucleotides with DNA nucleotides. It proofreads and removes primers. On the lagging strand, it fills the gaps between the DNA strands that have been discontinuously replicated. DNA ligase is represented by hairspray, which acts as a glue or sealant for the DNA that was added by the DNA polymerase I and III, and it does so without adding another nucleotide to the strand. In this event, the primers will not be removed and thus cannot be replaced with nucleotide bases. This also means that DNA ligase cannot seal the gaps in between the start and the end of the Okazaki fragments, resulting in a non-functional fragmented DNA.